Okay, so in this video we're going to talk about cell respiration and we're going to focus on glycolysis, the link reaction, and the Krebs cycle. So remember, the purpose of cell respiration is to take the energy in glucose, imagine represented by this line here, and to slowly release that energy through various methods, but slowly taking that energy out so it can be used for other things in the body, right? This is different from, say, the red line here, which is really just if you burned it all, you very quickly, use up all the energy. So that's what we're trying to avoid with cell respiration, is just a gradual pulling out of the energy. So the first stage is glycolysis. Now this is anaerobic, and it occurs in the cytoplasm. Okay, so there's two things. First, you need to invest 2 ATP molecules. Okay. So we got to spend a little bit here to make some more ATP. So what happens there is glucose is converted into hexobiphosphate. So basically, one inorganic phosphate from each of these ATP molecules is added to the glucose to make hexose biphosphate. Now, this makes the molecule unstable and it undergoes lysis. Okay, and it changes into triose phosphate, to 2 molecules of triose phosphate. Okay, and then there's a series of reduction reactions, phosphorylations, and ATP formation that it ends up with a molecule called pyruvate, which is still 3 carbons, 2 molecules of pyruvate from the 2 molecules of triose phosphate. Okay? So the first reaction is the reduction. So basically the idea here is an electron taken away from the triose phosphate and added to NAD plus to make NADH. Okay? And that happens twice. Okay? So high energy electrons move from triose phosphate onto NADH, or NAD plus, to form NADH and H plus ions. Okay. Second stage is a phosphorylation reaction. So we take an inorganic phosphate here and we add it to triose phosphate, creating triose bisphosphate. Okay. Then that allows us basically to, through another series of reactions, make 2 ATP molecules per 1 triose molecule. Okay. So those phosphates we added then are taken off to make ATP. So here, we make 2 ATP molecules. Over here, we make 2 ATP molecules. So our net gain here is 4 ATP molecules, but we had to spend 2 at the beginning. So we're down to 2 ATP molecules and 2 NADH H plus molecules. Okay, so we haven't made a huge amount of energy yet, but we're preparing to make more later. Okay, so then that pyruvate then goes into what we call the link reaction. Okay, in IB land, this is basically kind of combined with the Krebs cycle, but I'm going to just treat it as a different one for now. Now, this happens in the mitochondria, okay, in the matrix space, and this is considered aerobic. So to go from the cytoplasm to the mitochondria, right, the transport cost is 2 ATP molecules, right? There's ways it can get into the mitochondria, but for IB land, we're going to call it 2 ATP molecules. So remember, we spent 2 up here, or we had 2 extra here. So our net totals here, we have 0 ATP molecules because we had to spend 2 for the transport costs here. So this pyruvate is joined with this coenzyme A, CoA, and it undergoes a series of reactions. So here's another reduction reaction, right? So electrons from pyruvate are joined with NADH to make NADH plus, okay? And then there's what we call a decarboxylation reaction, okay? So carbon dioxide is removed. So we lose one carbon here, okay? And this is part of the carbon that we breathe out. That's where it came from, okay? Now remember, this happens twice, right? Because we have one pyruvate here, we have another one there. So it happens twice. So looking at this, we had 0 ATP. We didn't make any more ATP here, but we get a total of 4 NADH plus ions, right? So this is glycolysis and the link reaction. So we've gotten 0 ATP molecules and 4 NADH molecules, right? 2 from up here and 2 from the link reaction. Okay, then acetyl coenzyme A will drop that 2 carbon molecule into the Krebs cycle. And then coenzyme A is recycled, it goes and picks up another one. All right? Okay, so this happens in the mitochondria, this is in the matrix. Right? So then we go to the Krebs cycle. And again, this is also happening in the matrix of the mitochondria. And what happens here, right here, is the link reaction, right? So pyruvate comes in, comes out of acetyl coenzyme A, it's recycled, right? So here's the link reaction. So those 2 carbon molecules are joined with a 4 carbon molecule to make this 6 carbon molecule. And then we just have a series of different reactions happening here. I'm not so concerned with the names of them. That's not going to really help you. But there's a couple ones. So here we have another decarboxylation reaction. Here we have a reduction. Right? So we're stripping electrons away. Here, another reduction. And then here we have some ATP synthesis. Okay, notice there we go, 6, 5, 4. 2 carboxylation reactions. And these 4 carbon compounds are all they're not the same compound, they're different ones. So they're converted into a different 4 carbon compound. So from this conversion here, we have another molecule. We get electrons here. So FADH2 is like NADH. NADH is like like NADH, but think of it this way. This is kind of a little scooter. NADH is a truck. The scooter transfers a little bit less energy. The truck transfers more. That'll make sense later. But it's basically, like both of these are carrying electrons. Okay. And then a different 4 carbon molecule is made, and then there's another reduction reaction, and we're back to the beginning of this cycle, right? So as long as you keep feeding acetyl coenzyme A into this cycle will keep on turning. So looking at our net here, so now we have glycolysis, link, and Krebs. So we have a total, here we've made 1 ATP molecule. But remember, this is the Krebs bicycle, right? This turns twice because we have 2 pyruvate here, it makes 2 acetyl coenzyme A. So this spins twice, so the Krebs bicycle. Okay, so here we have 2, 4, 6. Okay, so we had 4 NAD here, so we have 10 NADH H plus ions. We have 2 FADH2 molecules and 2 ATP molecules. Okay. So again, at the end of this kind of cycle, when we've kind of worked through one glucose molecule, we're still we haven't made much ATP, right? We've only made 2. But we've made a whole bunch of NADH and FADH2, right? And that's going to be important when we get to the next stage, which is chemoosmosis ATP synthesis via chemoosmosis and that process. So that'll be the next video. So I hope this is helpful. Thank you.